Okay, so we are going to do our video review questions for the Acoelomate and Pseudocoelomate lecture, basically our second chapter in Principles 2. With this, we're looking at basically the groups we're going to look at in this section are going to be the sponges, the cnidarians, and the platyhelminthes, which are also known as the flatworms. Okay, so we're basically looking at those three groups in this chapter. Also nematodes and rotifers, so a couple of others as well. So we'll just kind of go through these questions. Don't have a ton of them, but a couple of these are fairly important for understanding the life history of some of these groups that we're looking at now. So our first question asks about the process of filter feeding in a sponge. And, this is really where, and I mentioned this also in the other lecture videos, this is where the sponges actually do exhibit some degree of almost tissue level organization, because the collar cells or choanocytes in the sponge are the ones that have a flagellum. They're the ones that create a water current to draw water in through the pores of the sponge, hence the name porifera. And because those cells are doing that sort of facilitation of feeding, that's kind of enough to put them in just enough of a gray area to consider these to be animals, right? Not really too much in the way of other specialization. We might look at a little bit around the osculum or the top part of the sponge for those that have sort of a structure like that. Those cells can also be somewhat cooperative, almost to the point of tissue level organization, because they can allow the osculum to either open or close depending on the needs of the rest of the sponge. The second question on here, we talked about in our first lecture or series of lecture videos, diploblastic versus triploblastic. Remember, diploblastic organisms are those that only have an endoderm and an ectoderm in the embryonic state, whereas triploblastic organisms have an endoderm, a mesoderm, and ectoderm, mesoderm, and endoderm if you're a triploblastic organism. So in this particular group that we're looking at now, of course, the sponges fit neither of those definitions. They don't really have tissues, and so therefore they don't have any germ layers. They're not diploblastic or triploblastic. Cnidarians are the only ones that are diploblastic, and everybody else in the animal kingdom is going to be considered to be triploblastic. All right, so we have one group, sponges, that don't qualify as either of them. We have one group that qualifies as diploblastic, and all of the other phyla of animals are going to be triploblastic. Okay, so because sponges do have a bit more organization, tissue level, than sponges do, we need to talk about some of the tissues they do have. Now, cnidarians are, even though they have tissues, the simplest organisms that have tissues, they really don't have very much, right? So they have nervous tissue. They have a lot of what's called mesoglia, which is kind of the jelly part, if it's a jellyfish or something like that. There's a few other cells that are specialized, definitely more so in sponges, but definitely nowhere near as much specialization as we see in the other groups that we're going to be looking at this semester. So there are a few tissue types, but really not a huge amount. In terms of intracellular versus extracellular digestion, this is a really important question. Again, sponges are unique in the fact that they exhibit intracellular digestion. I mentioned a few minutes ago, collar cells are going to move their flagella. That's going to draw water into the body of the sponge. But at that point, every single cell in that sponge is responsible for getting its own food. So each cell has to capture its own food particle, digest its own food particle within that cell. It's kind of like every cell for itself, even though there's cooperation to get the water and the food in the first place. All right, so sponges, porifera, they are intracellular digestion. They are the only ones that have intracellular digestion. All of the other animals, including ourselves, have extracellular digestion. So we take in food. It goes into a space of some sort, a stomach or an intestine of some sort, depending on the kind of animal we're looking at. And enzymes and so forth are secreted. Those enzymes and acids and things break down the food particles. The leftover bits, so the amino acids, the nucleotides, the monosaccharides, the fatty acids, those kinds of things are then absorbed in some way and then distributed throughout the rest of the body of the animal, right? So from sponges, we jump to cnidaria. Cnidarians and everybody else after them are going to have extracellular digestion, right? Food's broken down outside the cells, the leftover bits are absorbed and distributed, and the cells can from there have access to those nutrients. Okay, so all other animals have extracellular digestion. Okay, this next one's going to be a bit of a challenge because normally I would draw this on the board, but we're going to kind of talk through it. So for a hydrozoan, right, so hydrozoans are one of the 4 classes of cnidarians that we're looking at. We are looking at the, well, let's talk about the life cycle first. The life cycle of a hydrozoan is the most common example of a life cycle in the cnidaria. They're going to start off, we'll just start off at a zygote, product of a sperm cell and an egg cell fertilizing or joining together to form a zygote. Zygote's diploid. The zygote is going to undergo mitosis, and that's going to give rise to what's called a polyp form, P-O-L-Y-P, polyp. And that polyp form is the stationary phase of the cnidarians that have a polyp form. Most of them do. There are some that don't. We'll talk about that in a second. But the polyp stage is the sessile stage. It's the attached stage. Right? So what's going on in the polyp stage is generally going to be getting ready to produce the next part of the life cycle, which is the medusa stage. That's the part that kind of looks more like a jellyfish, probably what you're more familiar with when it comes to cnidarians, the overall basic body plan of a jellyfish. That is the medusa form. Right? So in most cnidarians, the polyps are going to give rise to medusa. The medusae, medusae with an E at the end of it, is plural, not medusas, but medusae. The medusae are still diploid, right? They haven't gone through meiosis yet, but they are the sexual stage, so that is where meiosis is going to happen. So these medusae are going to be dioecious, right? So that means that they're either male or female. They're generally not going to be both. They're not monoecious. They're not hermaphroditic. Every jellyfish is going to be either male or female. And therefore, when it undergoes meiosis, it will produce either sperm cells or egg cells, but not both, right? So then those sperm cells and egg cells get back together again. That forms the next generation of zygotes, and the life cycle is going to repeat. Now, I said that the hydrozoans illustrate this the best because their life cycle is, for most hydrozoans, something like 50-50, half polyp, half medusa, give or take. Now, there's exceptions here, right? So hydra, which you guys have drawn in lab now, or are drawing in lab now, Portuguese man oar is also a hydrozoan. They do things a bit differently, but for most of the hydrozoa, they illustrate more or less a 50-50 type life cycle with half polyp and half medusa, more or less. Now, if we look at the other classes of cnidaria, the scyphozoans, the true jellies, which I talk about in the videos, there's only like 200 species of those in the entire world. Most are saltwater, some are freshwater. These are going to be more so oriented toward the medusa stage. So in a jellyfish life cycle, they might spend 90% of their life in the medusa stage and only go into the polyp stage for a very, very short period of time. Often it's a very, very small polyp that's almost impossible to ever find because they're sitting on a rock somewhere, very tiny, cranking out medusa, and you really don't ever see them. Cubozoans, which are the box jellies, are even more extreme that way. They're more like, we'll say 95% medusa and 5% polyp. So they're even more oriented toward the motile stage of the cnidarian life cycle, which is the medusa stage. And then the anthozoans, which are corals and anemones and those kinds of things, they are going to be 100% polyp. So as far as we know, no anthozoans produce a medusa stage. They skip that part of the life cycle, and they tend to orient more toward an asexual type of reproduction because they are more of a sessile form. Now, corals certainly can be sexual. A lot of times, if there's a stressful event, a change in water chemistry, a storm, something like that, they can switch over to sexual, release gametes in the water, and have some genetic diversity produced that way. But for most of their life, when conditions are relatively stable, the anthozoans are going to be strictly polyp form in their life cycle. So you definitely want to make sure you know the 4 classes, who they are, what they look like, where they live, what their life cycle is like as well, okay? Okay, so number 7, we're now moving into the platyhelminthes, which are the flatworms. And so in the platyhelminthes, which is the phylum, there are essentially 2 subphyla within the platyhelminthes, which are the Turbularia and the Neodermata. Right? So turbularians are free-living flatworms. They are going to be out somewhere in nature, free-living, feeding on plants or feeding on detritus, which is just dead stuff that you find in the forest, for example. They're not really associated with a host, and they're not parasitic. Right? Turbularians are free-living. So they have eye spots. They have a typically more complicated digestive system because their food is not being digested for them by their host. On the other hand, the Neodermatans are the parasitic flatworms, which are then broken down into 2 classes, which are the Trematoda, which are the flukes, and the Cestota or Circummeromorpha, which are the tapeworms. So for those 2 groups, which are parasitic entirely, they're going to be much more adapted to that parasitic type lifestyle. So by that, they're going to have oftentimes more than one host. They'll have a host in the younger stage or stages for the flatworm, and then they'll have a stage for the adult form of the flatworm. That's actually fairly common. In parasitic flatworms, oftentimes they don't have any sort of eye spots, because why would they, right? It doesn't make sense to waste energy producing eyes or eye spots when you live inside the gut of a vertebrate of some kind. It's dark in there, so the eye spots really aren't going to do you any good, and so they don't have them. Tapeworms don't have them, flukes don't have them. Both of those groups also have much more simplified digestive systems, because especially for the tapeworms, they're just absorbing food that's already been broken down by their host in the stomach or the digestive tract in some place in that host. So for tapeworms especially, they actually, in many cases, don't even have a digestive system, because again, the nutrients they're getting have already been released from their host, and all they have to do is absorb them, right? Both groups also produce lots and lots and lots of eggs, hundreds of thousands of eggs, if not more, because getting to that next host is by no means a guarantee, and to have any chance of having at least one offspring even make it to the next stage or the adult stage, you really have to make a lot of eggs to make sure that at least one of them finds its way into a new host. All right, so trematodes and cestodes, much more, this is kind of like a bit of a paradox when we talk about evolution. We tend to think of evolution as producing adaptations that make organisms more complex, more structures, more fun kinds of things like a stomach and a liver and a complicated brain and kinds of things that we have. We like to think of ourselves as being sort of quote unquote highly evolved, but that's really kind of in the eye of the beholder, because a lot of times evolution produces simplicity. The tapeworms especially are extremely evolved. When we look at how they compare to the turbolarians, for example, they've changed a lot over evolutionary time, and they've become simpler. That doesn't mean they're not as adapted, doesn't mean they haven't evolved as much. In fact, they've evolved more. They've actually lost structures that their ancestors had. So try not to have like a bias against things. Just because they don't have structures doesn't mean they're not adapted to their environment. It doesn't mean they're not evolved, sorry, not evolved. Okay, number 8, we are looking at osmoregulation. And so osmoregulation primarily involves basically two things: maintaining water balance. And in the process of maintaining water balance, it's also going to be getting rid of metabolic waste. All right. So if we think about ourselves, we use our kidneys to regulate our water balance. If we're dehydrated, if we're overhydrated, our kidneys are hopefully going to take care of that for us. In the process of doing that, though, they're also going to help us get rid of metabolic waste that's been produced by our cells. It's been put into our blood. Our kidneys filter that out and help to get it out of our bodies. All right, so it turns out most organisms have some degree of osmoregulation that they have to undergo, because no matter where you live, there's an environmental challenge when it comes to water. Right? If you live in salt water, you live in a very hypertonic environment, you're going to tend to have ions coming in all the time. You're going to have water tending to leave all the time. And so your biggest challenge if you're a saltwater organism is just not becoming dehydrated because of that salty environment. On the other hand, if you're freshwater, you tend to have ions leaving your body, and you tend to have water entering your body, both of which, again, are not really necessarily ideal. So they face a very different set of challenges than, say, a saltwater type organism would. And for things that live on dry land, of course, dry land is it's dry. And so water loss becomes a very, very big issue on land. So when we talk about waterproof structures for terrestrial organisms, it's a little bit backwards from what humans typically think of when we think of waterproof. When we think of waterproofing, our home or having waterproof shoes or a waterproof jacket, we're trying to keep water out, right? But for most organisms, their problem is keeping water in, right? Except for freshwater organisms, saltwater and terrestrial organisms both have this issue of water leaving, and therefore evolution has produced a variety of structures to help keep water in as much as possible. Part of that's going to relate to question number 9 when we talk about a cuticle as well. All right, so question number 9, this is the first time we've seen a process known as ecdysis. And ecdysis involves shedding an outer cuticle in anticipation of growing a new cuticle. All right, so what is a cuticle? It's that waxy outer layer that surrounds some organisms like nematodes, for example. Arthropods do the same thing. So if it's an insect or a crustacean of some kind, or a millipede or a centipede or a spider, all of those things have a cuticle as well. But the issue with a cuticle, right? So it's on the outside of the body, but it's also non-living, right? That means that there's no mitosis going on. There's no expansion of cuticle. There's no growth of cuticle as the animal itself is trying to grow. So if the animal needs to get bigger, it actually has to get rid of its old cuticle, grow a new one, and then increase in size. And so ecdysis is the process of shedding the old cuticle to grow a new one to allow for growth. And it's not the same thing that reptiles do. That's a bit different. Their shedding is not completely unrelated, but it's a different kind of a story than what we're talking about here. So again, arthropods, nematodes, really commonly you'll find ecdysis in both of those two groups. And then rotifers are our other pseudocoelomate example in this chapter. So we have acoelomates, which were the sponges and the cnidarians. We have the pseudocoelomates, which are the nematodes and the rotifers. Okay, so rotifers are microscopic. They are always freshwater. Kind of interesting though, because they are multicellular and microscopic, right? So if you think of most things that we have to look at with a microscope because they're tiny, those things tend to be single-celled or maybe just a few cells. But with rotifers, they're made up of hundreds or thousands of cells, and yet they're still microscopic. So kind of interesting there. Normally we think of small things as being fairly simple that way. Rotifers definitely, though, are not that simple. Rotifers have what's known as a corona, which is a term for a ring of something. In the case of rotifers, the corona is a ring of cilia around the mouth that beats and therefore draws water into the mouth. Okay? So again, even though they're small, rotifers have a complete digestive tract. They have a tract with two openings, mouth and anus, as do nematodes, as do well, pretty much those two groups in this chapter that we're talking about right now. So cnidarians, remember, were they're going to have a single opening that serves as both the mouth and the anus that leads into their gastrovascular cavity. I didn't mention that before, but the gastrovascular cavity in a cnidarian is kind of their like one size fits all organ for just about everything they need to do. It's for feeding and digestion. It's for gas exchange. It's for waste removal. It's for releasing gametes. Everything kind of happens with the digestive or the gastrovascular cavity in a cnidarian. So one opening for them, two openings for every other organism that comes after them. And of course, sponges don't have a digestive tract. So, mouth and anus don't really apply if you're a sponge. So again, rotifers, tiny, have a corona, live in fresh water, essentially are kind of like filter feeders, but they're also pseudocoelomates. All right, so we're going to cut this off here. Of course, if you have any questions, please be sure to let me know. Normally, I would do a bit more drawing and things on the board, but at this point, we're just trying to stay on track as much as we can. So keep me posted with questions, and I'll do my best to answer them. And we'll see you in class.